Chapter 12, Segment 2, pages 378 through 386. Pheromones guide reproductive behavior in many species. Individuals are more likely to engage in sexual behaviors when steroid hormones of the gonads affect the brain. It is not absolute that they will, it's just more likely that they will engage in the sexual behaviors. When the gonads produce steroids to make gametes, they are informing the brain that the body is ready to mate. Pheromones are chemical signals that communicate information between animals to help coordinate reproductive activities. In rats, males have a sensory system that detects pheromones to activate male arousal. The vomeronasal organ, VMO, has specialized receptor cells near but separate from the olfactory epithelium, and they send their axons to the accessory olfactory bulb. The neurons here project to the medial amygdala because it depends on circulating sex steroids to maintain a masculine form and function. Lesions here will result in the inability to become erect that normally occurs around receptive females. The medial amygdala sends axons to the MPOA. The MPOA appears to integrate hormonal and sensory information, like pheromones, and it also coordinates the motor patterns for copulation. Chemicals that are part of the reproductive physiology also serve as pheromones. For example, in female goldfish, the hormone F-prostaglandin that is required for normal ovulation also is released into surroundings to stimulate male mating behavior and pattern. Rodents display many examples of probable sex pheromones. Pheromones in male mice urine accelerate puberty in young females. Females possess a particular type of vomeronasal receptor protein that can detect lordosis-inducing pheromones in tears of male mice, and females can identify an individual male by the mixture of pheromones in his urine. If the pheromones come from a dominant male, not a subordinate male, they can induce the birth of neurons in the olfactory bulb and hippocampus of the female. The Bruce effect is the termination of pregnancy caused by the odor of a pheromone in the urine of a male other than the one who impregnated the female. This was first observed in mice. Pheromones also function as a way of conveying messages between individuals of the same sex. Most is a mating season for elephants, in which elephants release a pheromone-laden liquid from glands on their temple, just behind their eyes. Adult males release frontalin, and this secretion signals the male's rank, and this will attract females. Pubescent males release a substance that has a honey-like odor, and it may attract bees and birds, but it is not strong-smelling like the adult males, and it helps the younger elephants avoid aggressive encounters. The hallmark of human sexual behavior is diversity. Alfred Kinsey constructed a set of standardized questions and procedures to obtain information for samples of the U.S. population, in which he categorized by sex, age, religion, and education. He eventually published many surveys on sexual behavior of American males and females. The results of these surveys concluded that nearly all men masturbate. College-educated people are more likely to engage in oral sex than non-college educated individuals. Many people have engaged in sex with another individual of the same sex at one time, and a stable portion of the population preferred such sex over heterosexual sex. After the publishing of Kinsey's study, physician William Masters and psychologist Virginia Johnson studied human sexual behavior by making behavioral and physiological observations of people engaged in sexual intercourse or masturbation. Of all mammalian species, including most non-human primates, most males will mount a female from the rear. Among humans, though, face-to-face positions are more common. A wide variety of sex positions exist, and many couples vary their positions from session to session, or even within a session. It is this variety in reproductive behaviors, rather than the different reproductive anatomy, that distinguishes human sexuality from that of other species. Another difference between humans and other species is that humans can report their subjective experiences of sexual behavior, especially with regards to orgasms, which are the brief, pleasurable sensations experienced by most men during ejaculation and most women during copulation. In Masters and Johnson's original conceptual model of human sexuality, there are summarizes of the typical patterns of both men and women, and it consisted of 4 phases. The first phase was increasing excitement, followed by plateau, then orgasm, and resolution. During the excitement phase, the phallus, which is the penis in men, and the clitoris in women, swells with blood, making it erect. In women, the parasympathetic activity during this phase causes changes in the vaginal blood vessels, and this results in the production of lubricating fluids that facilitate intromission. Stimulating of the penis, clitoris, and vagina during the rhythmic thrusting along the intromission may result in an orgasm. An orgasm in both men and women is accompanied by a series of contractions of the genital muscles. This will aid men in ejaculation and contractions of the uterus and vagina in women. The sexual responses for women and men had basic similarities but differed in important ways. Women show a wider variety of commonly observed copulatory sequences, at least 3 according to the model of Masters and Johnson. Men have only 1 basic pattern. Most men, but not most women, have an absolute refractory period following an orgasm, which essentially means they cannot have a full erection and orgasm until some time has passed. Women can have multiple orgasms in succession. Functional imaging of men and women's brains during sexual activity suggests that brain circuitry associated with an orgasm itself is similar between the two. There are very different networks that are active during sexual activity prior to the orgasm. Research has found that basic sex drive is greater in men than in women, and this is reflected by more frequent masturbation, sexual fantasies, and pursuit of sexual contacts. In men, there are closer links to sexuality and aggression, ranging from differences in sexual assertiveness to the most extreme case of rape. With regards to women, emotional components and cognitive factors play crucial roles in their sex lives. Women have a flexible sexuality that adapts to new experiences and situations over time, and most women place more emphasis on sexual intimacy within the context of a committed relationship. According to this finding, emotional intimacy and desire play a crucial role in the initiation of sexual responses, and after a sexual encounter, a combination of both emotional and physical satisfaction affects the likelihood of future sexual activity. Hormones play only a permissive role in human sexual behavior. Boys who fail to produce testosterone at puberty show little interest in engaging in sex unless they are given an androgen supplement. These males, as well as men who have lost their testes, have made it possible to experiment and show that testosterone does stimulate sexual interest and activity, as well as an increased sense of energy in men. In rats, additional testosterone has no effect on the drive of mating, and as a result, there is no correlation between the amount of androgens produced by the male rat and his likelihood to copulate. This is true in humans too, but less testosterone is sufficient to restore behavior. And despite popular beliefs, there is no correlation between systematic androgen levels and sexual activity among men, given that they have some level of androgen. Women sometimes experience sexual dysfunction after menopause by reporting a decreased sexual desire and the difficulty in achieving comfortable intercourse. When postmenopausal were provided with low doses of estrogen and androgens, there can be a beneficial effect on the genital experience of sex and a modest increase in the woman's sexual interest. Several attempts have been made to see if a woman's sexual interest or participation in sexual behavior is affected by her menstrual cycle. In general, it has been found that such effects are small, and some studies failed to find a significant change in sex interest across the menstrual cycle at all. For many vertebrae, parental care determines offspring survival. Extensive parental care is needed for many species to ensure that their offspring survive. This is especially true for species with altricial babies, which are humans and songbirds, which are helpless due to their poor motor skills and underdeveloped sensory system. Precocial youth, such as ducks and horses, require the parent's care, but only in the form of food and protection. In these species, females alone may care for the baby. Pregnant rats lick their nipples to clean them for the pups, as well as expanding the amount of sensory cortex that responds to the skin surrounding the nipples, which prepares for the letdown reflex. Rat mothers, or dams, display 4 maternal tendencies: nest building, crouching over pups, retrieving pups, and nursing. Virgin female rats won't display these actions, and instead will find the smell of newborn pups aversive. The information of the odor of the pups projects via the olfactory bulb to the medial amygdala to the VMH. Lesions anywhere along this pathway will result in the virgin female to display maternal tendencies because she no longer can smell the odor. Virgin females that are habituated to the smell of newborn pups will also display maternal tendencies after some time, whereas the mother rat will display these characteristics immediately. In a parabiotic preparation, the effects of hormones on maternal behaviors in rats is demonstrated that two female rats were surgically conjoined and thus share a single blood supply, which ensures that any hormone exposed to one rat is exposed to the other as well. If one of these females is pregnant, but the other is not, the one that isn't pregnant but was exposed to the pregnant rat's blood will immediately display maternal tendencies. No single hormone alone is responsible for fostering maternal behavior, but rather a combination of several hormones, including estrogen, progesterone, and prolactin. There's plenty of evidence that hormones in pregnant humans also prepare the female to display maternal tendencies. The brain regions that control maternal behavior have a considerable amount of overlap with those that control sexual behavior. A brain region that regulates maternal behavior is the anteroventral periventricular nucleus, AVPV, and this is larger in females than in males, and it increases in size post-pregnancy. Men who are interested in babies show less of a spike in testosterone secretion in response to erotic videos. Sexual differentiation. Sexual differentiation is the process by which an individual develops a male or a female body and the behaviors that are associated with that gender. In mammals, the process begins before birth and continues into adulthood. Sometimes a person can be a mix of two sexes. Sex determination in mammals, every egg carries an X chromosome from the mother, and when it fuses with an X or Y bearing sperm, that will determine the way the body develops. An X bearing sperm will produce an XX female, and a Y bearing sperm will bear an XY male. The first consequence of sex determination in mammals is in the gonads. Early in development, an individual has a pair of indifferent gonads, glands that vaguely resemble testes and ovaries. In the first month of gestation, differential genetic instructions determine whether the indifferent gonads change into testes or ovaries. In mammals with the Y chromosome, a gene called SRY, which is sex-determining region on the Y chromosome, is responsible for the development of testes. The SRY protein causes indifferent gonad core cells to proliferate at the outer layers, and they develop into testes. In XX individuals, or an individual in which the SRY gene is ineffective, no SRY protein is produced, and the cells of the outer layer of the gonad proliferate more than those of the inner core, and as a result, an ovary forms. Gonadal hormones direct sexual differentiation of the body. For all mammals, the differences in bodies between males and females is a product of differential hormone secretion. Developing testes produces several hormones, and fetal ovaries produce very little hormone. If tissues are exposed to testicular hormones, the body will develop masculine features. If they are not, they will develop feminine features. The internal reproductive tracts are different in males and females, but the embryonic stage, all individuals have the precursor tissues for both systems. The early fetus has a genital tubercle that can form into a clitoris or a penis, and it also has two sets of ducts that connect the indifferent gonads to the outer body wall. The Wolffian ducts is the first type, and these, in males, they will develop into the epididymis, the vas deferens, and the seminal vesicles, while Mullerian ducts shrink until they cease to exist. The Mullerian ducts for females will develop into the oviducts, which are the fallopian tubes, uterus, and the inner vagina, and the Wolffian system will disintegrate as a result of this. Two testicular secretions are driving forces behind masculinization. The first is testosterone, because it promotes the development of the Wolffian system. The second is the anti-Mullerian hormone, AMH. This causes the regression of the Mullerian system. In the absence of testes and their secretions, the genital tract develops a feminine pattern as the Wolffian system regresses and the Mullerian system forms. Dihydrotestosterone, DHT, is a more potent androgen derived from testosterone. 5A reductase, which is an enzyme, and it converts testosterone into dihydrotestosterone. When DHT and 5A reductase come together in the genital skin, bipotential genitals are induced to form a scrotum and a penis. If the testosterone wasn't converted into DHT, the genitalia would only become partly masculinized. Sex chromosomes determine the sex of the gonad, and the exposure of gonadal hormones controls the sexual differentiation for the rest of the body. Changes in the sequence of sexual differentiation result in predictable changes in development. Some individuals only have one chromosome, a single X. Embryos with a single Y will not survive. In Turner syndrome, an apparent female has underdeveloped but recognizable ovaries. This is expected when no SRY gene is present. Sometimes an individual with XX chromosomes is exposed to androgens in utero, and depending on the degree of exposure, will be masculinized. Congenital adrenal hyperplasia, CAH. And in humans, this can be the cause of developing female to be exposed to excessive androgens before birth. The adrenal glands fail to produce enough corticosteroids, and instead produce large amounts of androgens. In XX individuals, the androgen levels produced are usually a moderate amount between a male and a female, and newborns have an intersex appearance. A phallus has intermediate size between a typical penis and clitoris, and has skin folds that resemble both a labia and a scrotum. Once an individual with CAH is born, excess adrenal can be corrected, but the sexual differentiation has already occurred, and this would require surgical modifications in order to correct it.